to your presentation.
Can you see my screen?
Yes, we can.
Full screen, right?
Yes.
All right, good afternoon, everyone. Thanks for tuning in. It's my pleasure to talk about the flow cytometry core and some of the cutting edge technologies that we have to offer nowadays. So just to give you some numbers, we are a team of 6, so we have 5 staff working to help you, 2 managers, Luke and Wei. We have Steve and Xiaotian, who are a PhD level scientist, and Jesse, who is a staff associate. We are located on 2 sites, the 3rd floor of the ICRC building and on the 17th floor of the PNS building. We have 9 instruments, 5 analyzers that are operated by users, and 4 cell sorters that are operated by our own staff. In addition, we also have a Aura flow, spectral flow cytometer that belongs to the Human Immune Monitoring Core, but which we operate. So we serve approximately about 200 PIs at Columbia. So our services include daily calibration and maintenance of all instruments, troubleshooting if you have any issue during regular hours. As I said, we operate all the sorters ourselves, and that's included in the fees. We provide free consultation and training, and we also help with data analysis if needed. So for those of you who are not too familiar with flow cytometry, I just want to go very quickly about the type of the arsenal of toolkits that we have available to analyze cells. It goes way beyond just identifying cells or subpopulation of cells using specific markers. You can look at the expression level of number of functional molecules and the percentage of cells expressing them. That includes ligands and receptors on the surface, transcription factors, intracellular cytokines, and number of other molecules. We can also look at signaling, receptor signaling by phosphoflow, looking at phosphorylation of certain molecule. For example, here, STAT5 phosphorylation under IL-2 receptor signaling, and you can see that depending on the cell type you're looking at, regulatory T cells can respond to lower concentration of IL-2 relative to CD8 T cells, for example. We can look at cell divisions and cell cycle, and the cool thing about it is you can look at the cells, how many cell divisions they are going through, but also you can specifically look at each of these generation and then compare the phenotype as they progress through cell division, and same thing with cell cycle. You cannot just only look at proteins, but you can measure RNA expression. That's not as common, but it's a technical prime flow. And that allows you, if you wish to correlate both RNA and protein expression. But also, if you don't have a good surface marker to identify your cells, and even you don't have any protein to mark the cell of interest, but you rely on the transcription factor, for example, and say there's no antibody available for that, and you can actually stain the RNA and identify the cell population of interest. Very common apoptotic assays, and also endocytosis assay, when you can look at uptake of labeled molecule by the cells of interest. Very cool also, you can do measurement in a real time, for example, along a time scale. For example, calcium flux, when you add a stimulus in your tube during the acquisition, and you can see the flux going on and down as you measure the cells in real time. You can also measure enzymatic activity, for example, aldehyde fluoroassay to look at aldehyde dehydrogenase activity. Or again, you can also look at the expression of reactive oxygen species expressed by subtypes of cells to see to what extent they undergo oxidative stress. So let's move on to the technologies. Probably most of you familiar with conventional flow. I'm showing here the high-end instrument we have had for a long time, the LSR. Well, you need to realize that this actually reaching obsolescence, and this instrument will disappear within 2 years. And so we are progressively going to need to replace them with a more sophisticated instrument like a spectral flow cytometer This guy here was released last year, and we're planning to acquire another one through an S10 grant application. So I'm going to show some data from this instrument, but I really, if you are not been contacted yet, I really encourage you, if you're a PI, or even if you're a student or postdoc, talk to your PI about joining us in this grant application to get this instrument in the next year. So the limitation of conventional flow is that for each fluorochrome that you're using, you use one laser to excite it, and you have only one sensor to measure the emission from that excitation. As opposed to that, in spectral flow, for each fluorochrome, you can measure the emission from up to 5 different lasers. And for each emission, you can detect it in multiple sensors that correspond to different wavelengths. So you can basically go through the whole spectrum, and you get a very precise profile for each fluorochrome emission spectrum. So this kind of instrument is actually not very complicated to use. It's a very smooth transition going from conventional to spectral flow cytometry. You get to keep your program of choice for analysis, FlowJo, FCS, this works just the same. And you also get to keep your current panel. So if you use the LSRs up to 18 colors, you get to use the exact same antibodies, and you can add more to them. And if you want to add more, you have a much wider variety of choice, because now you can use fluorochromes that are similar, and could not be used before in combination in conventional flow. But now you can use both of them. And this is just an example here, APC and Alexafluor 647. Typically, they would be excited by the red laser. So this is here, the spectrum, emission spectrum and the red laser. You see it's very similar. But now if you look at the emission after excitation by the other lasers, you can start to see some differences. So based on that, the signal from these two dye can actually be distinguished very easily. If the cells express both of these fluorochrome, they can be distinguished as double positive cells, shown here, for example, NK cells, or NKT cells. You can see double positive cells staying with APC and Alexa 647. This was not possible before with conventional flow. Another feature is very unique to the Aura, is the ability to subtract autofluorescence from large cells. So if you use with stromal cells or cancer cells, these have very high fluorescence. And if on top of that, your signal is very low, then it can be very difficult to detect the positive from the negative signal. And here's an example, I guess, a prime flow assay, looking at the expression of low expressed genes in macrophage cell line. If you compare the profile, this is here from a 3-laser Aurora, the unstained versus the stained, the emission spectrum is very similar. It's only at the level of a few sensors that you see a difference. And subtracting that signal from the overall stain sample, you're able to actually now to separate what's really positive from what is negative. Another example here is from a lab here at Columbia, where they've been trying to analyze cells that express, so these are gut epithelial cells that express the YFP fluorescent reporter. And by conventional flow, it's almost impossible to distinguish the negatives from the positive. Going on Aurora, it gets a little bit better. The negatives, you can see them here, positive, and the dead cells. But again, everything is so bright, so strong that everything's pushed to the right. But with autofluorescence extraction, that you get a nice separation of the negatives and the positive for further analysis. So now when you can go up to 40 colors, that allows you to do a very extensive analysis of some human sample. For example, here, this is a panel of 39 markers plus live dead stain applied to human blood. And this very unbiased analysis, there's no gating needed to go up to that stage. You can isolate, you can see all these subpopulations shown here on the left. These are meta clusters. So these represent cell population or subpopulations. And there, when then you apply the expression of the markers of interest, you can identify where your monocytes are, the myeloid cells, NK cells, for example, B cells, or T cells. And within each population, you can see where the activated versus memory or naive. And so this is a totally unbiased approach and has some advantages. For example, I just want to show you here the analysis again on human spleen, trying to look at NK cells. And if you do unbiased analysis by T-SNP plot, then you can easily again see your B cells based on CD20 expression, your T cells are there, the CD8s here, and these are the CD4. But for NK cells, one of the more commonly used markers is CD56. And sometimes if you don't have this issue of the staining, you might have a problem knowing where to place the gate. So here you can see that CD56 in this case was actually found in B cells and in T cells as well. So there's something wrong there. If you look at the whole population, there seems to be a cluster here that express higher level, and it could be cells here that a few cells that express CD56. But there's a more unbiased way to clean up these data if you actually further subgate on the cells that express this higher level of CD56, which may be background. And then you can see better the cells of interest, and you can see they are clustering here together. And you know the NK cells because they express either the immature marker CD117 or the mature marker CD94 that is very specific to NK cells as well. So you can verify, you can look at additional markers in your panel. This is a 25 marker panel, for example, expression of NK P molecule like 4680, which is more highly expressed in a more mature population. And you can also look at other markers, some of them which you would not expect to be expressed in NK cells like HLA-DR, but you can have some interesting surprises to see other molecules like CD11C being expressed by NK cells to a substantial level. Now I want to show another study done in the Reshev lab. These are data from Rodney Macedo. In their lab, they're actually trying to characterize T-cell subset based on a chemokine receptor profile. And the idea behind it is you want to see if you can inhibit the migration of specific subset of T-cells, either to improve response to tumor, for example, or to, for example, if these are regulatory T-cells, or to inhibit specifically inhibit or and such to change the ratio of different subset of T cells based on chemokine receptor. So these are data gated on either CD4 and CD8 T cells, and you can identify population that express, for example, CXCR6 or CCR5 or CXCR3 or both, and among the negative CCR7. You can identify all of these population on a T-SNP plot here. And then for each of these population, you can see where certain markers are expressed. So these markers can be other chemokine receptors, they can be transcription factors like TBET, EOS, they can be cytotoxic molecules. And then you'll be able to see what subsets express them in very quick fashion. Of course, you can represent these as a heat map. So again, looking at the relative expression of each of these markers in those panels, looking at subsets of T cells that have been identified based on chemokine receptor expression alone. So these data have been done on PBMCs with the intent in the near future to apply these analyses to cancer infiltrating T cells. But to show the proof of concept that there are indeed within tumors T cells that express chemokine receptors with different patterns. These are single cell analysis performed on melanoma samples from humans. And you can see that indeed cells that are expressing only CXCR6 or these chemokine receptors with various combinations. And then finally, I want to show a last example, this time coming from the Faber lab. This work done looking at gamma delta T cells, which is a population of T cells that is not really well characterized, particularly in humans. It's not many of them. And as you might be familiar with the work by Donna Faber, they do very systematic analysis of immune cells across various human lymphoid and non-lymphoid tissues. And here we're looking at the expression of various markers on gamma delta T cells isolated either from the lung as a type of non-lymphoid tissues and lung draining lymph node as lymphoid tissues. And you can see that the ones in the lung are actually have more of a effector memory phenotype. They express more of these markers, whereas in the lymph nodes they don't. They also express higher level of tissue residency markers, like CD69 and CD103, which express at lower level in draining lymph node. And more remarkably, we can see co-expression of the cytotoxic molecules of perforin and granzyme B, but only in the tissue, but not in the lymph node. There are also differences in markers of terminal differentiation, for example, CD57, which is found expressed in the lung, but not in the draining lymph nodes. And in contrast, the ones in the lymph node express high level of CD28, which suggests that the restimulation in the tissue might be less dependent on co-stimulation like from P7 molecules. Just like you do TSNE, you can also do principal component analysis on across different samples from different tissues. And depending on what tissue they come from, you can see that these samples from different donors cluster very well, depending on the tissue origin, whether it's from the lung, from intestinal tissues, or from lymph nodes in general. So I will stop here with the technology. Just want to say a few words about where we are now in regards to COVID-19. The flow core is still operating as an essential service. We have minimal staff on site, but we bring the staff as needed. We operate one sorter at a time per site, and we ask people to reserve the instrument at least a day ahead so we can make sure that each instrument is calibrated still every day and operator present for sorting. If you have COVID-related studies, we work with you to make sure that these studies can happen. We have a number of users who reached out to us. I just want to mention prominently the Faber lab is already starting to do analysis of immune cells from COVID patients. We've gone through the protocols with EHS, and this is all now approved. And we're also going to do some sorting of live plasmacytes from convalescent patients with the whole lab. And for this, we have been able to upgrade one of our sorter room in ICRC 301A. And this is being given restricted access, enhanced operating protocols, negative pressure, and gowning area. So don't forget, we provide free consultation and training. So if you're interested in our services, we offer a variety of instruments that fits your needs. I show you a wide range of functional assays that can be done on single cell. And come to us with your questions and challenges, and we'll be happy to help.
Great. Thank you so much, Remi. That was terrific. And it's good to see that you're functioning and working away during this difficult time. We have a question here. I'm curious about the time scale for the calcium signaling measurement. I had not seen that before.
The time scales?
Yes. Alison, do you want to unmute yourself? And
yeah, I was interested in the fact that you could actually see the change in the calcium signal. And I know that the signal code can be in like 15 minutes, but I wasn't sure if that was the time scale of how you could see that signal. Because you said you add the signal, you said you added this, like the stimulant to the cells while they're being analyzed?
Yes.
Okay.
So you can access the tube while the cells are being acquired.
Okay.
You can add a stimulus, and then it's just a matter of minutes. You can start to see the signal.
And then what if you did something like to block that signal? You could see that too. I'm guessing.
Yeah.
Probably. Yeah.
Everything is happening in real time.
Cool. Okay. Thanks.